Good morning, good afternoon, and good evening to everyone logging in from different parts of the globe. Thank you all for attending today's webinar sponsored by the American Thoracic Society's Assembly on Allergy, Immunology, and Inflammation. My name is Anukul Shenoy, and I'm an assistant professor of microbiology and immunology and pulmonary and critical care medicine at the University of Michigan Medical School at Ann Arbor. I will be moderating today's discussion titled 2025 Assembly on Allergy, Immunology, and Inflammation Abstract Scholarship Awardee Presentations Part 2. This webinar will include 4 fantastic Abstract Scholarship Award winners that were awarded the Abstracts Awards at the ATS International Conference in 2025. And our discussion will be joined by an expert panelist, Dr. Benjamin D. Medoff, MD, Division of Pulmonary and Critical Care Medicine, Center for Immunology and Inflammatory Diseases, Mass General Brigham, Howard University. We ask that each presenter speaks up to 10 minutes, and we will have approximately 2 to 3 minutes after each speaker for questions and comments. For the audience members in the group, a reminder that if you have any questions, please raise your hands using the raise hand icon at the bottom of your Zoom screen, and I will call upon you to unmute yourself and you can ask your questions personally to the speakers. The session will be recorded and posted on the American Thoracic Society website, and a recording link will be sent to all the registrants. So without further delay, we will start with a presentation by our first speaker today, who will be Dr. Divya Bhatia, PhD, Instructor of Immunology in Medicine, Weill Cornell Medicine, Division of Nephrology and Hypertension, Joan and Sanford I. Weill Department of Medicine. Her abstract title is Mitochondrial Fusion Protein-mediated Interaction between Lung Macrophages and Type 2 Alveolar Epithelial Cells Prevents Lung Injury and Deleterious Kidney-Lung Crosstalk. With that, Dr. Bhatia, the screen is yours.
Thank you so much, Dr. Shenoy, for the introduction, and hello, everyone. I'll start sharing my screen, and before that, I would also like to thank the ATS and the AII Assembly particularly for this opportunity. I'll be talking about the role of mitochondrial fusion proteins in the kidney-lung interorgan interaction. So, chronic kidney disease is a global public health concern, and CKD-associated lung injury is underrecognized. Over 30% of the patients with the stage 5 CKD suffer with the restrictive lung dysfunction, and 11% suffer with obstructive lung dysfunction. The coexistence of chronic kidney disease with lung dysfunction not only increases the chances of organ failure, but significantly increases the rate of mortality. Therefore, it is critical to understand the mechanism of chronic kidney disease-associated lung injury. To do so, we are using 2 different models of kidney fibrosis, which is the final pathological manifestation of chronic kidney disease. The 2 models that we are using are unilateral ureteral obstruction, which is UUO, and Nathanial diet. We are interested in understanding how chronic kidney disease influences the immune microenvironment of the lungs, and particularly the role of mitochondrial fusion proteins, which are mitofusins, which are responsible for fusion of the mitochondria. We are interested in understanding how do the genetic deletions of mitochondrial fusion proteins in the type 2 alveolar epithelial cells versus macrophages impact their intercellular interaction, immune signals, and finally the systemic immune responses and the progression of kidney fibrosis. So we have observed that after 7 days of UUO-mediated kidney fibrosis, the expression of these mitochondrial fusion protein, mitofusin 1 and mitofusin 2, decreased in the lungs, as shown here. And we also checked the expression of these mitofusin 1 and 2 in the alveolar epithelial cells, the EpCAM positive, CD45 negative population, as shown here. And the expression of both the mitochondrial fusion proteins in the alveolar epithelial cells also decreased after UUO. While these mitofusins are suppressed in the lungs after kidney fibrosis, the fibrotic signaling in the lungs is induced, as we confirmed increase in the expression of these fibrotic and pro-fibrotic macrophage markers, including fibronectin, TGF-beta1, galectin-3, and arginase 1. So these findings suggest that the fibrotic signaling in the lungs is induced after kidney fibrosis. To understand the impact of chronic kidney disease on the immune microenvironment of the lungs, we next perform the immunophenotyping. We observed that the recruitment of leukocytes in the lungs, particularly the infiltrating CD11B expressing CCR2 and CCR4 high Ly6C intermediate monocytes, along with the Ly6G expressing CD11B positive neutrophils, increase in the lungs after 7 days of UO-mediated kidney fibrosis. We also checked the macrophage populations and observed that the number of interstitial macrophages as well as the monocyte-derived alveolar macrophages, as shown here in these flow cytometry dot plots, that the number of both of these macrophages increased in the lungs after UO. So these findings suggest that the recruitment of infiltrating monocyte-derived macrophages in the lungs increased after kidney fibrosis. To understand the macrophage-specific functions of mitochondrial fusion proteins, we next use these myeloid-specific mitofusin-1 deficient mice. We observed that the increase in the expression of these fibrotic markers in the lungs, as shown here, after UO was significantly suppressed in case of these myeloid-specific mitofusin-1 deficient mice, as shown here. To further confirm that there is a protection against kidney fibrosis-associated lung injury in case of this myeloid-specific mitofusin-1 deficient mice, we next check the lung macrophage-specific inflammatory responses. We observed that the lung macrophage-derived this lung macrophage-specific mitochondrial-derived ROS production increased after UO. However, the lung macrophages from these myeloid-specific mitofusin-1 deficient mice displayed lower inflammatory signals. These findings further confirm that myeloid-specific deficiency of mitofusin-1, we observed similar results in case of mitofusin-2 as well, that the deficiency of mitofusin in myeloid lineage protected against kidney fibrosis-associated inflammation in the lungs. We wanted to understand the reason for this protection. We performed further studies and we checked the expression of mitofusin-1. Interestingly, we observed that the alveolar epithelial cells from these myeloid-specific mitofusin-1 deficient mice displayed higher expression of mitofusin-1 compared to the alveolar epithelial cells from the control mice after UO-mediated kidney fibrosis. We thought that this compensatory increase in the expression of mitofusin-1 in alveolar epithelial cells in case of these myeloid-specific mitofusin-1 deficient mice might be responsible for exerting the protective effect against kidney fibrosis-associated lung injury in case of these knockouts. To further confirm these findings, we next performed the targeted deletion of mitofusins in type 2 alveolar epithelial cells using the SPC-CRE mice. We observed that in both of these single knockouts, the mitofusin-1 SPC-CRE as well as mitofusin-2 SPC-CRE, there was more infiltration of monocytes compared to the corresponding SPC-CRE control mice after UO. We also checked the number of the proportions of the macrophages, and in both of these knockouts, the proportions of interstitial macrophages were higher, while the tissue-resident alveolar macrophages were lower after UO compared to the corresponding SPC-CRE control mice. So these findings suggest that the recruitment of infiltrating monocytes in the lungs further increased in case of type 2 alveolar epithelial cell specific mitofusin-1 or 2 deficient mice. We next checked the lung macrophage specific functional responses and observed that the monocyte-derived alveolar macrophages and tissue-resident alveolar macrophages from these type 2 alveolar epithelial cell specific mitofusin-2 deficient mice displayed higher mitochondrial ROS production compared to the lung macrophages from the control mice after UO. And 3 of these macrophage populations, including the interstitial macrophages, start producing more galectin-3 in case of these type 2 alveolar epithelial cell specific mitofusin 2 deficient mice compared to the lung macrophages in the corresponding SPC3 control mice after UAO. These findings indicate that the lung macrophage-derived inflammation further increased in case of type 2 alveolar epithelial cell specific mitofusin 1 or 2 deficient state. So overall, our findings suggest that the lung macrophages during chronic kidney disease negatively regulate the mitochondrial functions of type 2 alveolar epithelial cells. And type 2 alveolar epithelial cells with impaired mitochondrial functions further promote the inflammatory responses by lung macrophages. And the lung macrophage-derived inflammatory signals exert systemic effects by promoting the migratory and inflammatory abilities of circulating monocytes. These inflammatory signals further promote the progression of kidney fibrosis. Due to lack of time, I could not be able to present these data from the systemic and the kidney and our cell culture work. But our findings suggest overall that the interaction between kidney and lung during chronic kidney disease is therefore bidirectionally deleterious. With this, I would like to thank my mentor, Dr. Mary Choi, for her support, all our collaborators and the funding agency for supporting this work, and of course to all of you for listening. Thank you. I'll be happy to take any questions later in the section.
Thank you so much for sharing this exciting data, Dr. Bhatia. We will open up this session for questions. Just for the audience, if you have any questions, please feel to use the raise hand icon at the bottom of your Zoom screen, and I will call on you to unmute yourself and ask the question to Dr. Bhatia yourself. We have Dr. Ma and Dr. Medoff who's with the first question. So Dr. Ma.
Thank you. Nice talk. I'm wondering, so your MFN1 expression is unique during the different type of macrophage, or is different, like the tissue resident macrophage, MOM, and the institutional macrophage? The expression is different during this different type of macrophage, or is unique?
So we have checked overall in not in all the subsets of the macrophages, but overall it is going in the same direction in the total CD64. But we haven't yet performed the sub-immunophenotyping because of the, this is an intracellular marker and this mitochondrial fusion protein. And in order to do the immunophenotyping for the extensive different panels, we have yet to do those this would be interesting to further confirm in the subsets of the macrophage population, but overall in total lung macrophages, it is going in the same direction. It is down. Thank you for the question.
Thank you.
Dr. Medoff.
Yeah, super interesting. And, of course, what I find especially illuminating is, we commonly think of the lung affecting the kidneys, and I think that makes a lot of sense. The lung is the biggest filter in the body, right? Your entire circulation goes through it at the same rate. So if there's inflammation in the lung, it literally bathes the blood in evil mediators that can affect other organs. Thinking about it the other way, though, the kidney only gets a small portion of the circulation. So I'm curious if and it's certainly interesting that these go down in the lung with kidney injury, and I really like your model. But so where do you think, I guess 2 parts. One is, what do you think is talking to the lung that's causing these changes? And I'm assuming it's something secreted in the blood, but if you have any thoughts. And then why did you focus in on these 2 proteins, these mitofusins, other than that they go down in this sort of situation? Was there some other sort of mechanistic reason that you kind of honed in on these and, found this kind of interesting relationship?
Thank you so much for both the questions. So in response to your first question, how do kidneys interact with the lungs? So in terms of after kidney injury, the tubular cells, they start producing the cytokines, including CCL2 and osteopoietin. CCL2 in particular is responsible for promoting the migratory functions of circulating monocytes. And these circulating monocytes, when they interact with the alveolar epithelial cells through their CCR2 receptor, they further establish this crosstalk. But the paper from Washington St. Louis group also published that AKI-associated ALI and the role of osteopentin in the intercellular interaction, inter-organ interaction between the kidney and lung also suggests the hypothesis with the role of macrophages carrying the osteopentin. And in response to your second question, why we selected the mitochondrial fusion proteins, so we earlier published using the Bleo model of fibrosis in Nature Communication that these double knockout, the type 2 alveolar epithelial cell specific double knockout, they show spontaneous lung fibrosis. And in our case, when we performed the immunophenotyping of the circulating system, we observed that the circulating monocytes from these knockouts behave differently. And in kidneys also, there is an induction of fibrotic signaling. After kidney fibrosis, there is more fibrosis in the kidney. So it's kind of crosstalk in a bidirectional way. So therefore, we chose this mitochondrial fusion proteins to further explore their role in the interorgan interaction.
Great. So do you think the CCL2 is getting into the circulation, binding to the endothelium in the lungs, since that's kind of the first sort of organ circulation it's seeing, and then that's bringing in the monocytes? Is that the sort of serving? Okay.
Yeah, interesting thing. So we earlier published in Kidney International that there is an induction of, this chemokine is induced in patients as well as in mouse model. And this chemokine binds with the receptor on the circulating monocytes, the CCR2 receptor on the monocytes, which then move to the lungs and establish this interaction with the epithelial cells in the lungs.
Very interesting.
Awesome.
Thank you.
I'm sure we can keep going with a bunch of questions and conversations. I'll move to the next speaker. Thank you, Dr. Bhatia, for this excellent presentation. Next up, we have Dr. Nathanial Tolman, M.Ed., M.D., PGY3 Internal Medicine, UPMC. He's a T32 postdoctoral scholar at the University of Pittsburgh. And Dr. Tolman's talk will be titled Lower Respiratory Tract, Candida albicans induces lung injury in mice and associates with lung injury endpoints in humans. Dr. Tolman, the screen is yours.
Thank you so much for the introduction, and thank you to the organizers and ATS and our panelists for the opportunity to talk about our work. So, as mentioned, my name is Nate Tolman. I'm a third-year medicine resident at the University of Pittsburgh, and our work is looking into the role of Candida in the lower respiratory tract. So the major question we're trying to address is whether Candida is a benign colonizer or pathogen in the lower respiratory tract of critically ill patients. As many of you know, Candida, and particularly Candida albicans, are among the most common organisms found in the lower respiratory tract in critically ill patients, particularly those who are mechanically ventilated. And it's traditionally been considered a colonizer, with the most recent Infectious Diseases Society of America guidelines recommending against routine treatment. However, we know Candida is also a pathogen at other mucosal surfaces, for instance, with oropharyngeal candidiasis or vulvovaginal candidiasis. Furthermore, recent studies have linked Candida albicans recovery from the lower respiratory tract in critically ill patients to things like longer hospitalizations, increased risk of ventilator-associated pneumonia, and even mortality. So we wondered whether we've been ignoring a modifiable risk factor during acute respiratory failure. So in order to address this question, we used a comprehensive translational approach. We analyzed critically ill prospective cohorts at 2 independent sites at the University of Pittsburgh and the University of Washington. These were mechanically ventilated patients in intensive care units who underwent lower respiratory tract sampling by internal transcribed spacer or ITS sequencing, as well as routine clinical cultures. We also developed a murine model of intratracheal Candida albicans installation to investigate its role in lung injury, and we utilized in vitro cytotoxicity and air-liquid interface assays in both murine and human lung epithelial cells. So first, to investigate the impact of Candida in the lower respiratory tract, we evaluated a subset of our critically ill cohort at the University of Pittsburgh who underwent ITS sequencing. And we separated these patients into those who had C. albicans dominance, which we defined as greater than 90% relative abundance of C. albicans, those with Candida species dominance, so greater than 90% non-C. albicans Candida species dominance, and then no Candida dominance. And strikingly, we found that those with C. albicans dominance were significantly less likely to be liberated from mechanical ventilation by 90 days when compared to the other two groups, with the number at risk shown below. To address this question in a separate way, we then looked at individuals with positive respiratory cultures and separated these individuals into those with normal respiratory flora, those with typical respiratory pathogens like Staph aureus, and those with yeast, which at our institution is Candida. Strikingly, those with yeast recovery had higher driving pressures than either those with typical respiratory pathogens or normal respiratory flora. And they had, in corroboration with our prior data, fewer ventilator-free days when compared to those with normal respiratory flora. And we were able to validate this at our University of Washington cohort as well, where those with yeast recovery had higher driving pressures and fewer ventilator-free days. So this suggests that Candida albicans in the lower respiratory tract associates with worse lung outcomes in critically ill humans. To sort of further address the role of Candida in the lower respiratory tract, we then utilized our mouse model, which it was C57 black 6 wild-type mice. We performed intratracheal instillation of 10 to the 6 CFUs of Candida albicans or vehicle control, and then we assessed lung injury endpoints at days 1 and 2. Candida-treated mice had significantly greater weight loss at both time points, and also had elevated levels of both IgM and RAGE, which is an alveolar epithelial injury marker when compared to vehicle control. They also had significantly greater lung injury by histology in a separate experiment, again suggesting that C. albicans alone is sufficient to induce lung epithelial injury in mice. We then looked back at our human data in those critically ill patients in our UPMC cohort with positive respiratory cultures. A subset of those individuals had host biomarker panel testing, and we found that those with yeast recovery had significantly higher RAGE levels in the lower respiratory tract as opposed to those with typical respiratory pathogens and a trend towards higher levels when compared to those with normal respiratory flora. To address how Candida may be impacting the lung epithelium, we wondered whether the yeast to hyphae transition, which is a known virulence factor at other mucosal surfaces, may be contributing in the lung compartment as well. To address this, we looked at BAL cytospins from our critically ill cohort and were able to identify hyphae in the lower respiratory tract of these patients. We then utilized a yeast lock C. albicans mutant strain, which is unable to form hyphae, as pictured here. And when we treated murine lung epithelial cells with the yeast lock mutant, we saw significantly attenuated lung epithelial cytotoxicity when compared to wild-type treated C. albicans. And when we looked at human lung epithelial cells at air-liquid interface, yeast lock treated cells had attenuated barrier disruption compared to wild-type treated cells. To take this a step further, we then went back to our mouse model and performed intratracheal instillation of 10 to the 6 of either our wild-type or yeast-locked C. albicans strains in the wild-type C57 black 6 mice. We saw that yeast-lock treated mice had less weight loss at days 1 and 2 and lower IgM and protein levels in their BAL, again suggestive of attenuated lung barrier disruption. Finally, we investigated the role of Candida lysine, which is a cytolytic peptide produced by Candida and encoded on the ECE1 gene, which is strongly induced during hyphal growth. And it's known to be a key effector of Candida's pathogenesis at other mucosal surfaces. We used 2 different Candida lysine mutants and a revertant strain with genetic reconstitution of the ECE1 gene. And we found that both Candida lysine mutants had significantly attenuated lung epithelial cytotoxicity, which was partially restored in our revertant strain. So in summary, I hope I've convinced you that Candida albicans associates with worse lung outcomes in critically ill patients, that it contributes to lung epithelial injury in both mice and human cell lines, and that hyphae are essential for this observed lung epithelial cytotoxicity and barrier disruption. While we find this evidence compelling, we are not recommending indiscriminate treatment for all patients at this point, but we do think this topic deserves further study. Some potential next steps would include investigating which patients are most likely to benefit. For example, those with high relative or absolute levels of Candida albicans, or patients with co-infection with specific pathogens like Staph aureus. Second, whether the timing of Candida is important for injury. The natural history of Candida in the lower respiratory tract in this patient population is understudied. And third, what are potential treatment strategies with options ranging from conventional antifungals to things like microbiome modulation? And with that, I'd like to thank all of my co-authors, and particularly my mentors, Drs. Bain and Kitsios, as well as my funding from the NIH, and of course the patients and their families, without which this would not be possible. And I'm happy to take any questions.
Thank you for that excellent talk, Dr. Tolman. We will open up the field for questions for the audience. If you have any questions, please click on the raise hand icon at the bottom, and we will have you unmute and ask your question personally to Dr. Tolman. But for now, we have Dr. Medoff with a question. Dr. Medoff.
That was a great talk and really super interesting. I don't think I get through a block on the MICU with not one of the interns asking me, they're growing Candida, do we need to treat this? And as you mentioned, usually the comment is, well, we see this and ignore it. I'm curious. I thought the mouse modeling is great, and I like the hyphae thing. I guess I have two questions. First is, do you know what Candida's stimulating in cells? Like I know, like some fungi hit the dectin receptors and things like that, and why hyphae may be more pathogenic, I think would be real interesting. I don't know if you know that. And the second is with the human data, which I give you plenty of props because that stuff is tough to do. But obviously we see Candida from oral culture, so just growing it in a respiratory culture. I'd be curious if you know how many of these had systemic or other sites growing Candida. And is there anything where if you grew it from the BALs you had or whatever, is that more pathogenic or more associated with worse injuries versus maybe just colonizing on a patient in their skin and mouth, et cetera?
Yeah, I really appreciate those questions. To your first about kind of the pathogenesis of Candida, and particularly hyphae, probably twofold. One is that, and again, a lot of this work is in particularly oropharyngeal candidiasis, where it's fairly well studied. But hyphae in particular seem to induce a stronger immune response in sort of the host through sort of various pathways, of which, as you mentioned, dectin is one possibility. And so there probably is a sort of stronger immune response, neutrophil influx, inflammation as a result. But it also does seem that Candida, particularly through Candida lysin, the hyphae, it strongly induces that gene and production of Candida lysin, which is directly cytotoxic to the epithelium as well. So probably twofold way that Candida actually induces epithelial injury when it becomes hyphal. And then the very interesting question about the human data. I don't know that we certainly, I don't think, have like a full study of other sites in these patients, but we may have, if we reviewed the EMR, a testing elsewhere that we could try to correlate those things to. So that would definitely be something interesting to look at. And then, I'm sorry, I'm forgetting the last part of the question now.
Well, it's just about that systemic component and whether, growing it in a culture obviously can be anywhere from if they're intubated, obviously but it could be in the trachea, it could be in the endotracheal tube, who knows? And just, is there, and when you had BALs, is there anything more predictive or associative if you get it from an actual BAL sample, presumably, sampling much more in the lower part of the airway tree?
Yeah. these were sort of, as you mentioned, mechanically ventilated patients. So it was either BAL or ETA samples. And so I think that's, probably part of the next steps too is probably better defining patient populations that may benefit from treatment and looking at things like, absolute quantity or, other things that may be present at the BAL that would signal, that treatment may be warranted. So definitely something we'd be interested in looking at.
We have a question from an attendee, Dr. Yuan Wang. Please feel free to unmute yourself.
Hi, can you hear me okay? Sorry, my video is off for some reason, but super interesting work, and I really commend the translational approach. My question relates to the role of pre-treatment with antibiotics, and I assume that most of the human data, patients would have received antibacterial antibiotics beforehand. And just curious, have you thought about how you might look at whether modulating the bacterial component in the lower airways and with different antibiotic regimens, how that might impact the behavior of the Candida and the effects on the on lung injury models that you looked at?
Yeah, that's a great question. We've in a sense looked at the opposite. We've looked at, and there's, decent data that Candida presence in the lower respiratory tract can predict or increases the risk, I should say, for VAP. And then we've actually done studies where in our mice we give Candida and then LPS to see if Candida potentiates lung injury. But I think it would be interesting to look at sort of the reverse, whether bacteria presence and the use of antibiotics would somehow change Candida's pathogenicity or characteristics. So that's a great question.
Great, thanks.
Thanks, Dr. Wang. I had a question as well. The Hyphal-lock version of the Candida, it doesn't produce the toxin?
So it's basically an overexpression of like a gene that keeps it in that sort of yeast-locked form. And the candidialysin is strongly expressed when it becomes hyphal, because that's when the ECE1 gene is upregulated. That being said, we haven't necessarily tested, whether there's any present or how much, but presumably because it doesn't form the hyphae, there's less than in a wild-type strain.
That makes sense. That's awesome. Thank you for that awesome presentation, Dr. Tolman.
Thank you.
Our next presenter today is Dr. Ruihua Ma, BS, MS. Dr. Ma is a research assistant professor of medicine at Northwestern University Feinberg School of Medicine. They are in the Division of Pulmonary and Critical Care Medicine, a Simpson Query Research Building. Dr. Ma is going to talk to us today about their work titled Vimentin Limits Regulatory T Cell Function and Delays Recovery from Viral Pneumonia. Dr. Ma, the screen is yours.
Thank you for kind introduction. Thank you, the panelists, and thank you, Dr. Maeda, for organizing this wonderful seminar. I'm Ruihua from Northwestern. It's my pleasure to share my research here. So we know this year's Nobel Prize was awarded to scientists for the discovery of regulatory T cell, also called Treg cells. So Treg cells is a type of CD4 T cell, high express CD25 on the surface and express the unique transcript factor FOXP3. So the classical role of Treg cell is to suppress immune response, thus to maintaining the peripheral silence and the inhibit the inflammation. Recently, it also been shown that Treg cells could promote tissue repair by generating the growth factors such as empathroglobulin and the keratin growth factor. So during the viral infection, because Treg cell could inhibit the inflammation and promote tissue repair, thus Treg cell promote the tissue recovery during viral pneumonia. So in our previous study, we show that the Vimentin is required for the flu-induced lung injury. As we can see that global knockout of Vimentin, the global knockout Vimentin knockout mice have better survival post-flu infection and have reduced inflammation and reduced lung injury. However, the specific role of Vimentin in Treg cell is unclear. So by immune staining, we found that Vimentin expressed in both human and mouse Treg cell. So in this study, we try to determine the role of Vimentin in Treg cell during recovery from viral pneumonia. So to determine the role of Vimentin in Treg cells, we use the adoptive T cell transfer colitis model. In this model, we adoptively transfer conventional T cell into regular mice, which lack immature T cell and B cells. So this will induce the mice weight loss and colitis development during the time. So in our model, we can see when we inject conventional T cell to regular mice, the mice lose weight over time and develop colitis in the colon. So if we give the Treg cell with conventional cell at the same time during the adoptive transfer, we found that sufficient number of wild-type Treg cells at a higher rate, Treg and T conventional 1 to 4, this Treg cell could successfully prevent the weight loss induced by the conventional T cell. And we can see that this Treg cell also rescue the mice developed from the colitis. However, if we decrease the wild-type Treg cell Treg cell amount, we decrease into 1 to 8 ratio, we found that at this concentration, wild-type Treg cell failed to prevent the mice failed to prevent the weight loss and failed to prevent the development of colitis. This suggests that sufficient vital Tregs are required for suppress the conventional T cell in vivo. Surprising, when we adopted transfer also the lower dose of Vimentin deficient Treg cell with the conventional T cell together, we found that this Vimentin deficient Treg cell could prevent the mice from weight loss at a lower amount of Treg cells. Also, these mice could prevent it from the colitis development. This result suggests that Vimentin deficient Treg cell have augmented suppressive function in vivo. So we wondering we want to study what's the role of Vimentin in Treg cell during viral pneumonia. So we generated the Treg cell specific Vimentin knockout mice by breeding FOXP3 with Vimentin frog mice. So then we intratracheal injection flew to Treg cell specific Vimentin knockout mice and later made control. And we can see that the knockout mice have better survival and faster weight recovery following flu infection. And when we analyze the lung, because as I mentioned that Treg cell could inhibit the inflammation, thus to protect flu-induced lung injury. So we analyze the lung 21 days post-flu infection. We can see that in the wild-type group, we still have strong lung injury as shown by HE staining, strong lung inflammation as shown by CD45 staining and flu quantification. And surprisingly, we also found the expansion of keratin 5 positive B cell-like cells in the wild-type lungs. So keratin 5 positive B cell-like cell is failed to repair the alveolar, thus is a dysregulated marker of lung repair. So however, in the knockout mice, we found a decreased lung injury, decreased lung inflammation, and have reduced keratin 5 positive cells. All this data suggests that Treg cell specific Vimentin deficiency protect from the flu-induced lung inflammation. So when we also corresponding when we measure the cytokine in the BAL sample, we found that all the inflammatory cytokine was decreased in the knockout mice. Also suggests the decreased inflammation in the knockout mice. So then what's the mechanism? Why the Vimentin non-Treg cell behavior better? So when we analyze all the Treg related suppressive marker and the transcriptional marker, we found that CD73 and Halos was upregulated. So we focus on Halos. So what is Halos? Hellos is a transcription factor which is essential for the Treg identity and stability. If we lose hellos, this Treg cell will become dysfunction, and the Treg cell lose their function and become the inflammatory T cells. So in our model, when we analyze the hellos positive Treg in the lung, we found that the Vimentin knockout, the Treg cell specific Vimentin knockout mice have increased hellos Treg cells in the lung. So further analysis showing that these hellos positive Treg cells have upregulated gene expression related to the Treg suppressive function and the Treg reparative function. So when we analyze the ARAG, immunoglobulin, which is critical for the lung repair, we found that the hellos positive Treg cells also have upregulation of hellos transcription level. So when sickle cell ataxic analyzed the hellos knockout and the wild-type Treg, found that in the ARAG promoter locus, the chromatin accessibility was much decreased in the hellos knockout Treg cells, suggesting that hellos is might a direct regulator of the ARAG expression. Interestingly, when we further analyze the hellos positive and hellos negative Treg cells, we found the upregulation of IL-18 receptor in the hellos positive cells. IL-18 receptor, so when we IL-18 receptor is one of the receptor expressed on the Treg cells, which is required for the immunoglobulin production. So when we confirm this by flow cytometry analyze the lung Treg cells in the wild-type and conditional knockout mice. We found that in the upregulation in the conditional knockout mice, we showing the upregulation of hellos. We also found these hellos positive Treg cells also express IL-18 receptor both in the baseline and the post-infection. So as this IL-18 receptor positive Treg cell could be stimulated by IL-18, induce the generation of immunoglobulin. So we culture wild-type Treg and Vimentin deficient Treg cells. Then in the presence of IL-18, 4 days later we measure the immunoglobulin. We found that IL-18 treatment induced the ARAG production in both wild-type and Vimentin non-Treg cells, but Vimentin non-Treg cell produce much more ARAG following the IL-18 treatment, suggests that the Vimentin deficiency augments the IL-18 hellos IL-18 receptor immunoglobulin signaling in the Treg cells, thus promote the Treg reparative function. So then mechanistically, why how Vimentin regulate the Treg function? So the interaction between Treg cells and antigen-presenting cells induce the formation of the immune synapse between these two cell types. At the same time, there is a protein complex called distal pol complex on the opposite side of T cell are formed. So the key marker of the distal pol complex, including the cell surface CD43 and the cytoplasma E3. So when we're doing the immune staining, we found that Vimentin is exactly located under CD43 expression and co-localized with E3, suggests that Vimentin is a key structure for the distal pol complex of Treg cells. So then we use the with afferent, which is a Vimentin with afferent, which is a Vimentin inhibitor, could bind to and disassemble Vimentin intermediate filaments. So when we treated the Treg cells with afferent, we found that disassemble of the distal pol complex structure, and so losing this distal pol complex structure correspondingly. So we found that the IL-18 receptor expression is changed because in wild-type Treg cells, IL-18 receptor polarized expressed on the distal pol complex. When we destroy this distal pol complex, we find the IL-18 expression being a unique expression around the cell surface. Functionally, disassemble this structure induce the upregulation of the repair factor immunoglobulin and induce the increased suppressive function. So this data suggests that Vimentin spatially sequesters the IL-18 receptor at the distal complex of Treg cells and suppresses Treg cell function. So in summary, this study found that following flu infection, they have secretion of IL-18 cytokine. This IL-18 cytokine binding to IL-18 receptor proteome and Helix proteome Treg cells, which further generates ARAG, promoting the tissue repair. At the same time, could inhibit the inflammation. So Vimentin could sequester this IL-18 receptor at the distal complex. If we disassemble Vimentin, we redistribute the IL-18 receptor around the cell surface and make it easy to capture their ligands and generate more ARAG. So finally, I want to thank all the amazing people who make this work possible. First, I want to thank my mentor, Dr. Karen Rich, for her constant support, guidance, and inspiration. And thank you all the lab members for their contribution. Thank you for the collaborators and for the funding resources. And thank you for your attention. I'm happy to take any question.
Thank you for sharing that exciting data, Dr. Ma. If there's any questions in the audience, please feel to raise your hand and I'll call you up to unmute yourself and ask a question. I saw Dr. Bhatia pop her hand up before Dr. Medoff, so Dr. Bhatia gets to start.
Thank you so much. Excellent presentation, Dr. Ma. I was just curious to know, did you compare the expression of CTLA between Helios positive and negative Tregs, the circulating as well as cell-specific expression of CTLA?
cDNA?
CTLA, the Treg specific receptor.
CTLA. So we do analyze the CTLA, but we don't find the difference.
I see.
Thank you.
Dr. Medoff?
Yeah, fantastic presentation and really very mechanistic work, which was great to see. And so kudos for that. I guess I'm really intrigued by this Helios marker, which normally I think of primarily being from thymic Tregs. I think the complexity of Tregs in the lung, especially there's innate Tregs that kind of are present there from the beginning, and then Tregs that are recruited that can have different functions. I'm curious if you think that these are Tregs in the lung that are upregulating Helios with the loss of Vimentin, or are these Tregs that you're thymic derived that are without Vimentin are maybe migrating more preferentially to the lung or something like that? It's a real interesting concept, and whether you've had any more details about the type of Tregs are there and their kind of presence in the lung either prior to the infection or during early in the infection.
Well, that's a really wonderful question. So we know that the Helios is the Treg have thymus Treg, have the inducible Treg, so peripheral Treg. So Helios indeed is thought to be a marker of the thymus Treg in the beginning. So in this study, we found that when we delete Vimentin, not only the lung Treg, also the spleen Treg was upregulation of the Helios expression. So whether we delete Helios induce the recruitment of Treg to the lung, I'm not sure, because we don't really differentiate whether it's resident lung Treg or the recruited Treg. So it's kind of difficult to define this type of Treg cells.
Have you looked like at the before infection, if there's more Tregs, or like in the early stage of like day 2 or 3 of the infection? Or
yeah, I need to show the data before. So in the baseline before the infection, we already showing the upregulation of the Helios positive Treg at the baseline. Yeah, we don't know. So I think it's kind of Vimentin is the basic regulation of the Helios expression.
Gotcha. And then my last question, and I'll let other people ask, is have you looked at viral clearance early on? Like,
yeah, that's
viral clearance is
that's a really awesome question. So we measure for this post-flu infection, which have really high abundance of the viral load. We don't find any difference between these two genotypes. So suggests Vimentin doesn't affect the viral clearance.
Very cool. Great.
Awesome. I had one question, Dr. Ma. Uh, the Tregs, because they are producing amphiregulin even in absence, like just by adding IL-18, they were increasing the production of amphiregulin. In vivo, do you think there's a role for like TCR, MHC signaling for these Tregs, or do you think they're more in an innate version of themselves where they are sensing the cytokines to get activated?
Thank you for your wonderful question. I think this is a really good question because it's not a TCR dependent, the environment generation. So this is a TCR independent way to generate environment. Also, it's specific to the tissue resident Treg, not the spleen Treg. So if you change spleen Treg with I-18, there are no environment generation.
Just because do you have any insights as to why? That's
So
pretty interesting.
I don't know. I think because that's kind of like the diff the tissue specific role of Treg.
Oh, that is pretty interesting. This is pretty awesome. Okay, so unfortunately, our fourth speaker was not able to get on call today. So we will be having only the three speakers, that Dr. Bhatia, Dr. Tolman, and Dr. Ma, that spoke today. So actually, that leaves us about 4 more minutes for people to raise their hands and ask questions. So if there's any other questions, or if the speakers want to ask each other questions, feel free to take the stage. Uh, I can essentially start with a priming question, I guess. I had a question for Dr. Tolman. Uh, I was very interested with the Candida lysine. Many bacterial pathogens also produce these toxins, and alveolar macrophages tend to be one of those first line of sensors for these toxins because they express so much of the NLRP3 and all these things. I was wondering, you talked about epithelial cells. Was there any insights into macrophage responsiveness to Candida in your models?
Yeah, it's a great question. I basically omitted half of the story for time. But we didn't look so much at macrophages, but actually at neutrophils, because they're very strongly recruited by Candida. And when we did sort of whole lung sequencing, I mean, it was just almost complete neutrophil activation recruitment signals. And we actually saw elevated neutrophil elastase in our human patients as well, and those with yeast recovery. Um, so it does seem that neutrophils play, an important role in both host defense and probably some inflammation. But we actually created a neutrophil depletion mouse model and then gave Candida, and we still saw a persistence of lung injury. And then it also disseminated to the kidneys and elsewhere, and they had greater mortality. So I think it's probably both that the neutrophils cause inflammation and epithelial injury, but that the candida itself also has a role in contributing to lung injury.
That is very interesting. That's pretty awesome. Okay, I don't see any more questions. I don't mind giving people 7 minutes of their day back. So, with that, I'd like to thank all our speakers for their excellent presentations. I would like to congratulate our speakers on their awards. Thank Dr. Medoff for serving as a panelist and coming up with really great questions that I hadn't thought of, and really keeping this discussion alive. I'd also like to thank all the audience members that joined and submitted questions, and thank the American Thoracic Society AII Assembly for sponsorship of this webinar. With that, we will conclude our webinar, and good afternoon, good morning, and good evening, wherever on the planet you are. Have a great day.